Hi, my name is Alicia Hernandez. Today is Tuesday, October 5th, 2021. This is week 5 and it's lab number 8. Okay, so prior to starting this experiment, I already washed my hands with warm water and soap, and I already cleaned my area with 10% bleach, and I already put on my PPE, and I already allowed the tubes of the phenol red broths to warm to room temperature for 30 minutes. And also I already placed the I already turned on the candle, and then I already label all my tubes, and then I have my alcohol with my 2 pipettes, and then I have my 6 Durham tubes, which I already put in alcohol, and I let them air dry. And so I'm gonna start. So I'm gonna, to each of them I have already the powder to the side. So I'm gonna grab this one, which is the staff one. So I'm gonna open it first and then sterilize the lip. And then I'm going to open up the mannitol, the Durham tube, and I'm going to put half on this one. Okay, so half of this. So then I'm going to sterilize the lip again. Then put the cap back on. So I'm going to shake the vial until the powder is dissolved. It looks like it already dissolved. So now I'm gonna grab the other broth, the phenol broth of the yeast, and I'm gonna repeat the same step. So I'm gonna open it up and then sterilize the lip. Then I'm gonna put the remaining of the powder, mannitol powder. I think that's it. So I'm going to sterilize the lip of the broth. Put the cap back on. Okay, so I already have here the Durham tubes from the alcohol that I let them air dry. So what I'm going to do is I'm going to grab the staff one, open it up, sterilize the lip, and then I'm gonna put this, it's called the Durham tube, facing down. I'm gonna sterilize the lip before I put the cap on. Take all the air out. So it has to be, the Durham has to be on the bottom. So after it's on the bottom, I'm going to open it up. Make sure the air is out, and then sterilize the lip again. And I'm going to repeat the same step for the yeast phenol broth. I'm going to open it up, and I'm going to pass the lip of the vial over the flame for the second time. And then I'm going to grab the Durham tube, and I'm going to put it facing down. Gonna sterilize the lip again. Then I'm gonna put the cap back on. And I'm going to get all the air out. Make sure that the dirham is on the bottom of the broth vial, which it is now. And then I'm gonna open it up. Oh no, I already did that. Okay. So let's see. So now I'm going to grab my pipette. I'm gonna sterilize it on the alcohol. And then make sure it's dry. So shake it to make sure it's dry. But before I do that, I need to grab I'm gonna start with the staff. I need to put 4 drops of these into the fennel red broth. So I'm gonna remove the cap. And then sterilize the lip over the flame. And then I'm gonna grab the same one. So this is the staff one, this is the staff one too. And then sterilize the lip. Okay. So again, I'm gonna grab my pipette, make sure it's dry, shake it to dry it. And I'm gonna put 4 drops of the active culture broth into the fennel red broth. Make sure it's completely dry. And shake it. So 1, 2, 3, 4. I don't think I'm gonna put this into the bleach. So before I put the cap on, I'm gonna sterilize the lip again. So I'm gonna repeat the next step for the yeast. Open it up. And then sterilize the lip. Gonna open up the methyl red broth. And sterilize the lip. So then I'm gonna grab my pipette, make sure I flush it with alcohol good to sterilize it. And I'm gonna shake it, make sure it's completely dry. So I'm gonna grab 4 drops of the Nutri broth of the yeast. And then put it into my fennel red broth. 1, 2, 3, 4. I'm gonna put it into my bleach. Before I put the cap on, I'm going to sterilize the broth, the lip of the broth. And then sterilize the lip of the fennel red in the broth. Let's see. Okay, and then so I already placed the pipettes into undiluted bleach. I'm going to repeat the steps, the same steps 17 to 21 on the other fennel red broths that I have labeled. And I already have them ready with the powder next to them. And then I'm going to incubate the fennel red broths for 12 hours. I'm going to wipe down my work area after I'm done with 10% bleach. I'm going to remove the used pipettes from the bleach and wrap in paper towels and dispose them in the garbage. And wash and return all other items from my kit for future use, and wash my hands thoroughly with warm water and soap. And that's it for today. Thank you.